concentration. After lunch, always a challenge. Okay, next topic we're going to have is very interesting. We're going to talk about food authenticity and profiling of foods. So there are going to be a few techniques here, including isotope ratio mass spec, which is very, very interesting. So let's give our attention to the next speaker, which is Vincent Myers.
Thank you, Lincoln. So far today, we've been speaking about chemical contaminants in food. But when we were designing this seminar, we thought, let's give our audience, our attendees, something a little bit different, something, a different kind of topic that's not necessarily chemical contamination. So we decided on a topic of food authenticity. I hope you enjoy the topic. So what is food authenticity? Food authenticity is the opposite of food fraud, when you're trying to fraudulently represent something. Food authenticity is about testing for the misrepresentation of either mislabeling or adulterating products using lower cost material. So usually food adulteration or food fraud has to do with economic gain, making a profit by giving some misrepresentation of a food. Here are some examples. Wine authenticity, when illegal sugars are added to grape juice, or the addition of sugars like high fructose corn syrup to honey, the addition of hazelnut oil to olive oils to make up more volume and make more profit. Mislabeling the geographical origin of things like wine, olive oil. The French, for example, are very proud of their wine, and if it says Bordeaux, it should come from the Bordeaux region of France. The Spanish are very proud of their olive oil, but sometimes oil that is sold, even though it says that it's Spanish oil, really may come from Turkey or Greece or some other part. The mislabeling of organic food products. Maybe it says organic food, in other words, has no pesticides, but it might. The adulteration of meat with cheaper species, expensive meat, by adding in cheaper species like chicken or pork. The European Union has really taken big steps to regulate authenticity, and they've come up with some schemes and systems. The first one is PDO, which means Protected Designation of Origin. This covers the agricultural products which come from a certain region. It also has another designation, PGI, Protected Geographical Indication. And this covers the agricultural products where one of the processes of manufacturing the product come from a particular region. They also have a scheme called TSG, Traditional Specialty Guarantee, to highlight the traditional character of a particular food. So as you can see, the Europeans have spent a lot of time on this. Just a minute, I'm trying to change the slide. And it's not changing. The European Union also has what's called the DOOR database. This DOOR database stands for Database of Origin and Registry. It has more than 1,200 entries. Many different countries participate, and it has a number of different foods like olive oils, wines, cheeses, honey, and beer. You can easily access the internet. You can do a Google search for DOOR database, and here it is on the internet. So this is live on the internet. And you can look up for many different products, just enter DOOR, and it will show you a list of different kinds kinds of products from many different countries. Sometimes the countries are not only from Europe, but they can be from Korea. You can do a search here under country. You can go to K. Where's K? KR is Korea. And you can just select that and do a search here. The internet here right now is a little slow, so I will not do the search, but this is easy for you to do. You can search products from many different countries. Okay, that's door. There are a number of food authentication challenges in the analytical laboratory. For example, many foods are chemically almost identical. And unique marker compounds are rarely found. More often than not, there are only small analytical differences, just like isotope ratios that we have to rely on. There's a large natural variability, fortunately for us, that are based on climatic conditions, the fertilizers that were used, the water that was used to irrigate the plants, the way the things were processed. So there are lots of natural variabilities, and we can use this in the laboratory to tell the difference between authentic products and fraudulent products. The techniques that we use must be able to distinguish between these small differences, and it's very good to use databases of authentic foods so that we can compare the fraudulent foods. Here is a list of several techniques that can be used. We can use infrared, Raman spectroscopy, NMR, or nuclear magnetic resonance spectroscopy. We can also look at elemental compositions with atomic absorption, ICP, ICP-MS. More complicated techniques are chromatography and mass spectrometry. And one of the more complicated and sophisticated techniques is isotope ratio mass spectrometry. So there are a number of choices to use in the laboratory. There are easier approaches. For example, DNA-based methods like PCR, polymerase chain reaction. These are used mainly for the adulteration of meat and meat-based products. So, for example, the addition of chicken and pork meat into minced beef, or the adulteration of foie gras, which is goose liver, with chicken, which is cheaper. There's also some work going on with the Chinese bird's nest that you see here. Here is John Gilbert and myself eating bird's nest a few days ago in Singapore. Thank you, Lincoln, for taking the photo. There are other techniques, immunology-based approaches, like ELISA, to detect the presence of soy in milk or meat, or the adulteration of expensive sheep milk or goat milk, which has less allergens, the adulteration of those expensive milks with cow milk. It's also good to use chemometrics technique. Chemometric software can be used to tell the difference of many different kinds of samples. You can typically take a large number of variables about samples, like their isotope ratio, or the elemental composition, the mass spectra, and other factors, and compare them, the fraudulent products with the authentic products. You can use multivariate analysis, and there are really two kinds of methods. There are supervised methods and unsupervised methods. The supervised methods are when you know what an authentic material looks like, when you know the characteristics of an authentic material. The unsupervised methods are when you have no knowledge about the origin or the factors of a particular food. These are just some terms and terminology for unsupervised methods and unsupervised methods. So there's ANOVA, MANOVA, these are variation analyses. There's principal component analysis, or PCA, and cluster analysis. These are different ways of categorizing an unknown food. You can also use supervised methods like linear discriminant analysis, partial least squares, and artificial neural networks to do supervised methods where you're comparing an unknown with an authentic food. One of the best ways to determine whether or not a food is authentic is by isotope ratio comparison. So here's an example of why we can tell fraudulent honey or adulterated honey from real honey. So isotopes exist in nature. For example, carbon isotopes exist naturally. C13 to C12 ratios, they exist, but they vary in nature. For example, when plants take on CO2 and they undergo photosynthesis, they manufacture sugar. And the kind of transition product that they make can either have 3 carbon atoms in the molecule or 4 carbon atoms. So some plants are called C3 plants and some plants are called C4 plants. Most plants in the world are C3 plants, but there are some that are C4. Honey is derived from plants that are C3, but corn is a C4 plant. It's important to know that the isotope ratio of C13 to C12 is different in C3 plants and C4 plants. So if you have honey, which has an isotope ratio here for C3 plants, and you mix it with high fructose corn syrup, which is a C4 plant, you will see an incorrect isotope ratio. And you can tell whether or not the honey is pure or whether it has been adulterated with cheap corn syrup. Another way to look at isotopes is through the hydrogen isotope, which is deuterium. Deuterium has an extra neutron in the nucleus. It's an isotope. So it's heavier than normal hydrogen. This heavier nature makes HDO heavier than water, H2O. The heavy water is more difficult to evaporate. It requires more energy to evaporate. So depending on the temperature of the region, you will have differences in this isotope ratio, HDO to H2O. Some scientists and institutes have made maps of this ratio depending on where they are in the world and have correlated that to temperature differences. This is important information when you're trying to determine whether or not a product comes from a certain region in the world or really comes from someplace else. An isotope ratio mass spectrometer can give very important information. An isotope ratio mass spec can look at many elements, for example, hydrogen and its isotope deuterium, carbon, C12, and its isotope C13, nitrogen, oxygen, sulfur, and many others. For example, I've seen papers on authenticity of caviar, which is fish eggs, based on the strontium isotope ratio, to tell whether or not it comes from a very expensive area like Finland, or whether it's from some cheap area, cheap caviar area, like the state of Washington in the US. And the information really comes in the number of significant digits that you can get from an isotope ratio mass spec. In order to get this kind of accuracy, you really need a very stable environment, which is what an IRMS gives you. So you can look for many different elements in an IRMS, but you have to consider how you want to enter the sample into the mass spectrometer. There are different ways of doing that. You can do it with a gas bench, which is for gases, or you can use bulk stable isotope analysis, and then there's also compound specific isotope analysis. I will compare bulk stable and compound specific isotope analysis in the next slide. So in bulk stable isotope analysis, you take your entire sample and you put it into a reactor. This is basically a combustion chamber at a very high temperature. And it burns everything up and turns it into things like carbon monoxide. And then the elements that are a product of this combustion chamber are separated in a GC column, then entered into the isotope ratio mass spectrometer. With this, you have all of the isotopes, all of the elements coming in, and you can determine the isotope ratios. The other way is compound-specific isotope analysis. In this case, we first do a separation of all the compounds. And one by one, we put them into a reactor, and then the isotope ratio mass spec to tell the isotope ratios. The benefit of separation of everything is you can get a chromatographic profile of the sample. So if you're analyzing wine or honey, you can take a look at the chromatogram first and then the isotope ratios. This gives you more information to determine whether or not something is authentic. Here's an example of the reaction. You have compounds that have carbons, hydrogens, oxygens. You put them into a very hot environment. This is the bulk isotope analysis. And then they produce CO, H2, C, carbon, and those are put into the isotope ratio mass spec, and that machine will determine the percentage of deuterium to hydrogen, the percentage of the isotope to the larger element. The sample comes through into the IRMS. It passes by a magnet, a very strong magnet, and then there is a multi-collector or a multi-element collector which will detect and determine the ratios of the isotopes. Here is an example of a system that we manufacture. It's called GC IsoLink. So this is compound-specific isotope ratio mass spec. Compound-specific because we separate out the components in the column, and then they go through a very high temperature oxidation and also another high temperature conversion, and then they go into the isotope ratio mass spec. This is good because, as I said before, it allows for the simultaneous determination of the elements and their isotopes, as well as giving a chromatographic profile of the sample. Lots of information. One example of using this is with tequila. Tequila is the national alcoholic drink of Mexico. The Mexican people are very proud of their tequila, but it must be authentic tequila. Tequila comes from the state of Guadalajara, the state of Jalisco in Mexico. In the state of Jalisco, there is a plant called the agave plant, and it only comes from here. Tequila is made from this agave plant, and it is tested for authenticity outside of Mexico City at the National Institute of Metrology. The reason why they test for this is because in the last 20 years, there have been fraudulent versions of tequila that come from other plants, not the agave plant. And oftentimes they come from countries that are very far away, like Taiwan. So the Mexican government tests for this, and they use an isotope ratio mass spec, and they have found that when they compare the oxygen isotope ratio and the carbon isotope ratio, the real tequila has characteristics right in this band, right around 11%, 018 to 016, and then about 11.3% of C13 to C12. If it is fraudulent tequila, made with sugarcane or some other plant, it will not fall in this area. It will fall someplace outside. So this is one way the Mexican government keeps control of authentic tequila. Honey is another commodity that is adulterated a lot. And the problem is that many unscrupulous providers add cheap sugars, like the fructose sugar, to honey. We can use an HPLC version of compound-specific isotope ratio mass spec and use an isolink system. Here's an example where we got the chromatographic profile of different honeys. You can see that the honeys chromatographically look different. But we also have information about the isotope ratios. We have the carbon isotope ratio information for different sugars, the glucose, the fructose. And this is very important data because we can determine if it is adulterated or not depending on these numbers. Remember, we can tell these isotope ratios of the fructose and the glucose because those are different peaks that we separate out before it goes into the combustion chamber. There are other uses, other applications for isotope ratio in authenticity. For example, we can do cheese. There are different ways to prepare cheese with lyophilization, some extraction, but it's always good to use additional techniques in order to determine real authenticity. So not only to rely on isotope ratio mass spec, because no technique is perfect, no technique is 100% certain, so you can also use ICP-MS. Just do a simple digestion, dilution, and into the ICP-MS. So this was done by a customer of ours, and they got the isotope ratios of different isotopes here and graphed them out, which gave some good information. But really the way to tell whether a cheese was really from Switzerland, because they call it Swiss cheese, or whether it was some cheese from Finland, or from France, or from Germany, is really by ICP-MS elemental analysis. So when our customer compared the amount of molybdenum and the amount of sodium, they found that Swiss cheese clustered right around here. And anything that had a different concentration of molybdenum and sodium fell outside of this area. So it wasn't only the isotope ratio information, it was also the elemental analysis that the customer could use to tell if something truly was Swiss cheese. There are other techniques that can be used. We've spoken before about infrared spectroscopy. Infrared like NIR-IR and FTIR, they're easy to use. They're very quick techniques. They require little to no sample preparation. The only problem is that they're not as sensitive as isotope ratio mass spec. This is our FTIR right here. One of our customers used our NIR-IR instrument to test for the geographic origins of olive oils. Remember, it's important to know the difference between olive oil from Spain, Italy, Greece, or Turkey, because they have different prices. And the customer used chemometric software and found when they entered in the NIR-IR data, the spectral data, they could find that different cheeses fell in different areas on the graph. So typically with this kind of chemometric software, it takes a little bit of work to train the system, to put in all the data and see where the data lies, And then after everything is set up, it's very easy to just take samples for routine analysis and see where the sample lands on the chart. So there are pros and cons of the different analytical techniques, depending on what you want. I talked about NMR, IRMS, chromatography, and elemental analysis. But depending on what you want to do in the laboratory, you will select a different technique. For example, here green means very good, yellow means so-so, and red means difficult. So if you want easy sample preparation, you may want to do NMR or infrared spectroscopy. If you want something very fast, good speed, then you use NMR or infrared spectroscopy. You can even use ICP-MS, which is very fast. If you want to use something that's very low cost, you may want to use an infrared spectroscopy technique. But if you really want to increase your probability of success and be more certain, not 100% certain, but more certain that you are testing the right, you have the right authentic product, then you want to use more reliable methods like isotope ratio mass spec or NMR. So which technique is most appropriate? It depends on what you want to do. Really, isotope ratio is considered a standard approach, but it is expensive. Organic mass spectrometry is gaining popularity in the lab. Elemental analysis is very useful to find out the geographic origins. And non-destructive techniques are very fast, very cheap, and require no sample preparation. These are near-IR, FTIR. And it's always good to use sophisticated data analysis techniques like chemometrics software. And Thermoscientific offers all of these solutions depending on what you want to do in the laboratory. That concludes my talk. I'm Samia.